Hi, my name is Malloy, and this is a presentation for the biotech class at the University of Connecticut. For this presentation, I will be discussing genetically modified GalSafe pigs. To start with the very basics, GalSafe pigs were created by a company called Revivicor, and the modification was approved by the FDA for food and therapeutic use in 2020. These pigs are genetically modified by homologous DNA recombination to not produce alpha-gal. An alpha-gal is a sugar residue that can be found on cell surfaces in mammals, excluding humans and some non-human primates. And we will get to why alpha-gal is relevant in just a moment. First, I'm going to go into a little more detail about how these pigs were engineered. Alpha-gal, which is actually galactose alpha-1,3-galactose, is produced by the GGTA1 gene. And it was discovered that inserting a specific recombinant DNA construct knocks out this gene by homologous recombination and therefore gets rid of the production of alpha-gal. So to create these pigs, the recombinant DNA construct was inserted into fetal fibroblasts, which were then transferred into enucleated oocytes, and those were then transferred into surrogates, essentially utilizing somatic cell nuclear transfer. 5 hemizygous pigs were produced, meaning they still had one active copy of the gene, and they were then bred and produced homozygous pigs that did not produce any detectable amounts of alpha-gal because they had 2 copies of the recombinant DNA construct. And this modification was passed and can continue to be passed through conventional breeding. So you may be wondering what the significance of alpha-gal is and why pigs are being modified not to express it. And there are a couple of reasons. First, I'm going to talk about something called alpha-gal syndrome. This syndrome is a tick-borne allergic condition that is passed by the saliva of Lone Star ticks. So for instance, somebody is bitten by one of these ticks that has alpha-gal in its saliva. It enters the body and the immune system creates antibodies for it. Then when the person is exposed to alpha-gal again, primarily through red meat or any animal products that may contain it, they can have a potentially life-threatening allergic reaction, which typically occurs about 3 to 6 hours after consuming the alpha-gal. This condition is not incredibly well known or researched as it is rather new. However, case numbers are increasing. From 2017 to 2021, the CDC recorded about 90,000 potential new cases, and they were seeing increases by about 15,000 cases per year. So going back to the modified pigs, it would be beneficial to have a meat alternative for people with this allergy that otherwise would not be able to consume pork products. Also, pigs are genetically modified for a variety of applications in human medicine. In fact, the same company that produces the GalSafe pigs also produce pigs for human organ transplants. And it's been hypothesized that the alpha-gal can be responsible for transplant rejection even if the person receiving the organ does not have an alpha-gal allergy. So it is a relatively simple modification that can further ensure the success of using pigs for human medicine. As mentioned previously, the modification in these GalSafe pigs have been approved by the FDA. It's actually the first approval of its kind in that it's the first intentional genome alteration in pigs that has both a food and therapeutic use. So far, there's been no real public pushback, most likely because it is still new and not something that people are very aware of, but that could easily change, especially as alpha-gal syndrome becomes more widespread. However, the FDA has done their due diligence ensuring that these pigs are safe for human consumption and medical use. They have found that aside from the alpha-gal, modified pigs and regular pigs are indistinguishable. They've also made sure to analyze multiple generations of the modified pigs to ensure that that modification remains stable and that the alpha-gal was still undetectable in those further generations. Overall, this is a very useful genetic modification that has the potential to have substantial impacts, whether that be on people with the alpha-gal allergy or in ensuring the success of organ transplants. Thank you for listening.